Hello, everyone. Thank you for attending today's webinar titled Synthetic DNA Technologies Enable Antibody Discovery and Optimization. My name is Jordan Wong, Vice President of Antibody Discovery at LakPharma, and also today's moderator. I'm thrilled to host today's webinar. Before we get started, please feel free to submit any questions to our speaker at any time by clicking on the Q&A button located on the toolbar at the bottom of the window. If we cannot get your question today, we will respond via email. Please note this webinar is recorded and the recording will be sent to you 1 week after. LakPharma is delighted to announce a strategic collaboration with Twist Biopharma, in which we can offer Twist's proprietary antibody discovery and optimization platforms presented today, including Twist custom libraries. Customers of Twist Bioscience will also have access to LakPharma's antibody discovery and development services. Our speaker, Dr. Aaron Sato, will go into more details later about the Twist libraries. And now, I would like to introduce today's speaker. Dr. Aaron Sato is the Chief Scientific Officer at the Biopharma division of Twist Bioscience. Prior to Twist, he served as CSO of LakPharma, and I personally had the pleasure of working with him to discover and develop novel antibody therapeutics for our clients. Dr. Sato also oversaw discovery research functions, both as Vice President of Protein Sciences at Surrozen and previously as Vice President of Research at Sutro Biopharma. He also held director-level positions at Oncomed and Dyax Corp. And now we turn over to Dr. Sato.
Thanks so much, Jordan. Again, my name is Aaron Sato. I'm the CSO of the Biopharma vertical within Twist Bioscience. And today I'm going to be talking to you about how we utilize our synthetic DNA technologies to allow us to discover antibodies as well as to optimize them. One of my idols is Benjamin Franklin, not because he was a politician or an author or an artist, but he was also an inventor. He created all kinds of cool things like the lightning rod, for example, that basically enabled people to do impossible things that they couldn't do before. And so I also aspire to do the same thing, but in the field of antibody engineering. Can I create new technologies that basically enable people to do things that they haven't been able to do before in the antibody engineering field? And in particular, when I think about new drug targets, I often apply kind of three main criteria that my boss at Dyax, Clive Wood, actually instilled in me a long time ago. And those are, first, does the target have a therapeutic rationale for going after it? Does it actually fulfill an unmet medical need? And then finally, is there actually technology to address that target and actually create something that could have an impact on it? And so what I really love to find are targets or even classes of targets where the first two are very clear, but the third one is actually, it's actually enabled by creating new technology that I might be able to actually have a hand at. And in addition to creating new technologies to, again, make the impossible possible, I also really love to collaborate with others to potentially enable some of these groundbreaking technologies. And hopefully by today, you'll see that the technology that we have at Twist really enables the next generation of the discovery of the next generation of antibody drugs for pharma and biotech. One book that I read recently that I continue to really love and also can suggest for others to read is called The One Thing. The reason why I love this book is that it basically outlines that the best companies and the most successful individuals really understand their one purpose in life, their one mission. And then they try to maximize the potential of that one thing. And so I'm going to use this as a framework for my talk today, where I'm going to basically walk through Twist Bioscience's one thing and how we then utilize that for all kinds of different fantastic purposes, in particular in the area of antibody drug discovery. So our one thing at Twist Bioscience is our ability to write DNA on silicon. So again, in a traditional setting, you would typically make oligos in a 96-well plate as shown on the left. Utilizing the Twist technology, we can actually print millions of oligos on a silicon chip about the same size as that plate. And we also can print them in, again, we're making discrete oligos here, print them in these clusters of 121, up to 121 discrete oligos in these individual clusters on the surface. And we can actually elute off those clusters and actually utilize those oligos to make genes, which is one of our major products that we have at Twist. But we also can make large pools of oligos and utilize them for example, making DNA libraries, which I'll talk about in a second. And again, this is a really groundbreaking, game-changing technology that enables us to make libraries that are very just unprecedented in the industry compared to what people have been able to do in the past. So how do we do that? So again, as I said, utilizing the technologies, and the one thing that separates Twist from others is their ability to make oligos in large pools. And so if you think about an antibody variable domain, like a heavy chain or a light chain, and then think about how we can make large pools of discrete oligos, you could think of each of those pools as basically being the information that would encode for a specific collection of CDRs in that variable domain. So shown here is just an example where I've created 3 different oligo pools that encode for CDRs 1, 2, and 3. And then I've stitched them together seamlessly to create a variable combinatorial library for that specific domain. The reason why I'm building libraries in this way is really differentiating is that I can, because I'm making each oligo separately in a pool, I can make sure that those oligos, for example, match the natural human repertoire. Whereas people in the past have tried to do this by making degenerate oligos, either through mixtures of oligonucleotides or mixtures of trinucleotides to somehow mimic what's seen in nature. Here, I can actually encode for the exact sequences that are in the natural human repertoire. In addition, because I'm making each oligo separately and then pooling them together, I also can remove sequences that may have liabilities in them. So I can actually remove potential developability problems up front before I even build the library. And so I can remove isomerization sites, cleavage sites, deamidation sites, even glycosylation sites from those natural human sequences up front before I actually make the library. And then also finally, we also can do kind of unique things like if I want to encode specific motifs in those sequences as well. Again, you could never do that with a degenerate oligo here because I'm making each oligo separately and I'm pooling them together. I can do fancy things like that. So just to summarize, the oligo pool technology really enables us to make unprecedented libraries that people haven't been able to do in the past, and they have a lot of advantages over traditional NNK and trim-based approaches in the sense that we can remove all liabilities from our sequences as well as potentially match the natural human repertoire and also potentially contain sequence motifs if we choose to. So now I'm going to transition and talk about 2 technologies that we've created that are enabled by this oligo pool and library technology. And so to apply this, I wanted to think about ways at which I could use it to solve something that has been really difficult for me to crack in the past. And that issue for me was actually discovering antibodies to G protein coupled receptors. So folks in my industry know that it's very difficult to discover antibodies to GPCRs. And so it actually reminded me of my very first job back at Dyax. Actually shown here on the right is my ID from Dyax. I pride myself on, I actually look pretty similar to the way I looked back then, but even with that said, so way back when I was at Dyax, one of the first projects I worked on was a collaboration with a large biotech company where we were hired to basically utilize our semi-synthetic phage display library to discover antibodies to CCR5. We put the target through all kinds of different strategies with peptides and overexpressing cell lines, and snippets of the GPCR itself, and we just couldn't crack it. And at the end of the day, we had to give up on the project. And looking back on it, I think our issue with this particular project was that our library just didn't have the content in it that allowed us to discover antibodies to this important target. So thinking back to how we could potentially crack this problem, we really need to have 2 different technologies to enable it. First, we have to have a library that has these antibodies in it, which again, I think was probably my issue back in the day. And then second, we also have to have technologies allow us to create and present these targets. And so I'll talk today about how we're using overexpressing cell lines, but certainly there's lots of folks in the industry that are making all kinds of different variations on expressing and presenting GPCRs to libraries such as this. So I'm probably preaching to the choir that GPCRs are an important class of targets. Actually, if you look at all the targets that are validated mostly by the small molecule field, about half of them are actually GPCRs. So the small molecule field has actually shown us that this is a really rich class of targets. But I think a lot of us would agree that antibodies would be favorable to small molecules in the sense that antibodies, of course, oftentimes are more specific than small molecules and potentially could give us a longer duration and half-life. But again, there hasn't been enough innovation in this space to really allow us to find antibodies readily to this important class of targets. And so our approach is, can we create a fully synthetic human antibody phage display library that we can pan against any GPCR and hopefully pull out these unique and very valuable antibodies against this class of targets? So what was our approach? We actually took a look back and actually found this paper in 2014 by Liu et al., where they actually took a crystal structure of CXCR4 bound to a loop peptide. And what they did in the paper is they actually took that loop peptide and had the idea of, can I actually graft that peptide into a known antibody structure and see if I can now bind to CXCR4? And that's actually what they showed. They took a bovine IgG, which, as many of you know, has a very long heavy chain CDR3 loop. And actually, for the longest versions of these bovine IgGs, they actually have this knotted structure at the top. So they actually took that knotted structure off, grafted on their loop peptide, and found, as shown on the right, that the antibody now binds to CXCR4 transfected cells and also has function in a migration assay shown in the bottom right. So this is the concept that we originally took on for building our own library. Can we create, rather than a bovine scaffold, a fully human scaffold that has a very long heavy chain CDR3 stem that can accept motifs that are known to bind to GPCRs? But rather than just use one motif, we want to use upwards of 100,000 different motifs derived from all kinds of different content that I'll talk about in a second, and make it a general purpose library that we can use to select for antibodies to any GPCR that we want. So where did we get those motifs? We extracted them from, of course, known chemokine GPCR interactions. And again, we're just taking small snippets of these proteins that are known to bind directly with the GPCR. There's also a whole family of different helical peptides that are known to bind to GPCRs, as well as loop peptides, like in the previous example. In many cases, the N-terminal domain of many GPCRs actually folds back and interacts with the second extracellular loop of the GPCR, and so we took some motif content from the N-terminal domain. And then we, of course, even though finding antibodies to GPCRs is difficult, there actually are a fair number of GPCR antibodies out there, and so we took a lot of H3 content from those antibodies as well. So after a huge amount of work, we basically looked at several different fully human germline scaffolds, and as well as some long heavy chain CDR3 stems, and eventually homed in on a family of heavy and light chains that could accept long heavy chain CDR3 sequences, and then we grafted in these motifs to create a fully human library that had these specific GPCR motifs grafted into it. And so at the end of the day, we now have a library of over 10 billion different antibodies with these motifs grafted into it. This was a huge endeavor that we did in collaboration with Distributed Bio to create this library. And so today, we can now pan this library against any GPCR target we want and pull out specific functional antibodies to that target. And I'll talk about one specific proof of concept study that we did with the library today. So in terms of presenting the GPCR to the library, we created overexpressing cell lines that had several different tags for us to assess both cell surface expression with an N-terminal flag tag, and then a GFP tag on the C-terminus to look at for sorting for overall high expression. And so this is what we use initially for our proof of concept, but as I mentioned before, we are actively seeking out partnerships with GPCR companies that have other enabling technologies that can present these GPCRs to our library. So for the discovery work that we do, we typically, of course, do cell-based pannings using our GPCR library. As I mentioned before, it is a fully human library in phage. It's also an SCFV format, and we have an overall diversity of greater than 10 billion. We do about 4 or 5 rounds of panning, and then we assess the clonal enrichment of those clones from round to round using next generation sequencing. And then at the end, we'll do Sanger sequencing to pull out all of our unique clones from the rounds 4 and 5 output. Because we're Twist and we can synthesize hundreds of genes on demand, we typically will reformat all of our uniques to full IgG, and then we'll scale up that DNA and do transient transfections to produce and purify those antibodies. And then we'll assess their binding and functional potency. So for our first proof of concept, we used an overexpressing cell line that expressed GLP-1 receptor, which is involved in diabetes. Shown here is just the NGS data from that panning that we did. And so shown on this slide is basically a frequency analysis comparing the clonal enrichment as we go from round to round in this panning. And what you're trying to look for is, do I see greater and greater frequency overlap of specific heavy chain clones as I go from rounds 1 to round 5? And as you can see here, what we're seeing significant clonal enrichment, particularly in rounds 4 and 5. And those are the rounds that we eventually picked to pull out our unique clones that potentially bind this target. So as I mentioned, we made over 100 IgGs from this panning, and of those, about 15 of them actually bound to the GLP-1R overexpressing cells. Shown here are 8 of them, where I'm showing the titration curves for binding to GLP-1 overexpressing cells compared to the CHO parental BRCA brown cell. And so you can see we're seeing EC50s in the range of single-digit nanomolar all the way to hundreds of nanomolar. And of these 8 antibodies, we also assessed their function in a cyclic AMP assay. And so what we did is we did a titration of the known ligand for GLP-1 receptor. This is in a DiscoverX assay. And then we compared the activity of that activation with the peptide in the presence of a high concentration of the antibody. And what we saw for all of these 8 antibodies, they all inhibited or pushed the EC50 activation curve in this assay to the right. So they were all either antagonists or negative allosteric effectors in this particular assay. Finally, another thing that was really encouraging about the output from this library is the actual sequences and of the hits that we identified. So of the 15 that we found, many of them actually had the GLP-1 peptide motif. So we were actually able to extract out motifs that made sense based on the target that we panned on. And so all the ones shown in yellow are actually motifs derived from the natural GLP-1 peptide. In addition, we were also able to pull out motifs for GLP-2, which even though GLP-2 is known not to bind to GLP-1 receptor, in the context of our antibody, we were actually able to pull out motifs that were similar to GLP-1 peptide. And then finally, as part of the library, we also built in a whole collection of long, fully human heavy chain CDR3s. And so the ones shown in white are actually examples of just fully human heavy chain CDR3s that don't actually have a motif. One question I often get asked is how developable are these antibodies, because they have these really long heavy chain CDR3 sequences. And I'm just showing you a couple examples, but in general, the antibodies express well, they're monomeric, they're not prone to aggregation, they're not sticky. And so as part of any discovery campaign, we oftentimes will do these developability assays as a QC check for the antibodies that we're creating. But in general, we're seeing really good behavior of the antibodies coming out of the library. So as I mentioned, we're actively seeking GPCR technology partnerships, and again, working in collaboration with LakPharma. In many cases, the clients that would come to work with LakPharma would most likely have their particular GPCR target of interest maybe already expressed in a cell line, or maybe have access to soluble protein. But if not, we have a lot of collaborations that we're establishing with outside companies to potentially enable us to get these GPCRs to enable a discovery campaign. So the second technology that I'll talk about today is how we can utilize the same DNA library technology utilizing oligo pools to optimize antibodies. So in this case, we can take an antibody derived from any source, whether it's murine all the way to fully human, and again, we can put this into our software, which has a large NGS database behind it of up to 12 individual people. And what the software does is it basically looks at the CDR sequences of your antibody and then compares that to the NGS database and then pulls in all of the CDRs that are similar based on a defined mutational criteria that you set. So for example, I can tell it, give me all the CDR sequences that are within 3 mutations of my parent sequence, and then it'll look into the human NGS database to find and see if there's any CDR sequences that match that mutational criteria. And then once we have that data, we can then create a library from it and basically pan and screen it back against the original target. So to show you the power of this technology, I'm going to show you again a proof of concept study that we did with a PD1 antibody. This is a PD1 antibody derived from a Cambridge antibody technology library. It's a fully human antibody, and it's also a lambda antibody. Shown here on this slide is just an alignment to the closest related germline. As we build these libraries, we actually have to select a single germline scaffold to graft this diversity into. And so oftentimes we'll just use the fully human germline scaffold, but of course, if the client decided to use a scaffold with some mutations in it, we certainly could do that as well. Again, shown here, we can input a number of criteria into the software as we optimize the sequence. Again, as I said, we can define the mutational scanning distance from the reference antibody compared to the NGS database. We also can define how many people we want each individual CDR sequence to show up in. And so oftentimes I'll say at least 2 people have to have that specific CDR sequence. And then finally, we also can tell the software to actually explore germline sequence within that same defined mutational space as well to give us greater diversity around our specific CDR sequences. So at the end of the day, we basically, again, input the antibody sequence. The software gives us a whole series of oligo pools, which are shown here for this specific example. We order those oligo pools from Twist, and then we create a DNA library from that. We clone that back into phage, or we could use, again, any other display platform. And then we pan and screen that back against the original target. And the overall timing for this project takes about 4 months. So shown here is just the panning NGS assessment of that in this particular POC study. Unlike a lot of other affinity maturations that I've been involved in the past, which typically their diversity collapses really early, maybe in rounds 2 or 3. For these particular libraries, we often find that we have to pan them for upwards of 4 or 5 rounds to see significant clonal enrichment. And again, that's just a testament to the overall diversity of these libraries, and they really allow one to very effectively screen mutational space around one specific lead. And so I find this is a kind of definitely a next generation approach to creating optimization and affinity maturation libraries that allows one to really drive forward the best specific lead. Which another thing that we assessed as part of the NGS analysis is when we looked at the clonal enrichment of these clones and then compared that back to their ELISA signal, we found that we really did have to push it out to later rounds in the sense that the most enriched clones actually had the highest ELISA signal. So again, showing us that to really obtain the most specific and highest affinity clones, we really had to thoroughly select and pan the library out to later rounds. And so then after we did that, we then did Sanger sequencing of all our positive clones. We reformatted them all to full IgG, and we made upwards of 100 different antibodies. And then we screen them on our CAR-Terra platform, very similar to what LakPharma has as well in terms of their internal capabilities. So shown here is just a whole series of different IgGs that we assessed on our CAR-Terra platform. Shown in green is the parent antibody, which has a really fast on-off, and with an affinity of roughly 325 nanomolar. Shown in purple are the two marketed approved commercial antibodies, pembrolizumab and nivolumab. It's just comparative controls. And then shown in red are actually several antibodies that have comparable or maybe even better affinities compared to what's actually on the market today. When we actually stack up all those sequences in terms of their relative affinity from lowest with the parent on the bottom to the highest affinity at the top, we can really see how the CDR sequences are changing in response to this shuffling of natural human diversity in the library, and how specific mutations are popping up that are driving towards higher affinity. And so you can see at the top many of these changes that are very apparent through the sequences that we're getting that are coming through with the optimization that we're doing against PD1. And so at the end of the day, we actually had 6 antibodies that had higher affinity or comparable affinity to nivolumab and pembrolizumab. Another cool thing we like to do is just look at the phylogenetic tree of these sequences, and as you can see, we have a large number of different sequence families. And another cool observation that we found is that more than 90% of the clones also had unique light chains that were never repeated. So again, a testament to the diversity of the original starting library. We're actually able to have a lot of diversity in our overall, even the final sequences that we identified. Finally, another observation that we made, which was really cool, is that when we actually looked at these antibodies in a functional assay, so this is a Promega PD1, PD-L1 functional reporter assay, we compared the EC50s in this assay to the overall affinities that we determined, and we found that there was actually a linear correlation between the two in the sense that the highest affinity clones also had the most function. So since we had 6 antibodies that had higher affinity than Nivo, we actually had 6 antibodies that also were more functional. So at the end of the day, again, collaboration with LakPharma, of course, we're open to licensing these libraries, but also partnering the 2 base technologies that I talked about today. We're also creating some, through the POCs I just talked about, we're also generating a lot of leads through that work. And so those may be available for licensing or partnerships down the road. So looking ahead to the future, I talked to you today about our GPCR library and our TAO library, but we're also going to be rolling out a whole series of different antibody phage display libraries that they'll be in different formats, either Fab format or SCFE format or even single domain format. And again, all of those will be available through our partnership with LakPharma. So just to wrap up, again, starting with my idol, Benjamin Franklin, thinking about ways we can make the impossible possible. At Twist Biopharma, our one thing is our ability to print DNA on silicon, and that enables us to make unprecedented libraries. And I've shown you 2 examples of how we did that with our GPCR library as well as our TAO platform today. So looking ahead to the future, my goal is to basically create and build a library of libraries. So someday I hope that I'll actually have a deep well plate in the freezer that'll actually have 96 different libraries that we can basically pull out and utilize. And again, through our collaboration with LakPharma, they would have access to all of those libraries that my group would create. And then finally, I would just like to thank the team. We have a team of about 12 people in South San Francisco that's basically created all these libraries that I talked about today, as well as done all the proof of concept work that I've shown. And so again, I'm really proud of their achievement and accomplishments as well. And thank you for your attention.
All right, thank you, Dr. Sato, for your presentation. Since we don't have a lot of time left, we're gonna, for the Q&A, we're just gonna help answer 1 or 2 questions. We're gonna launch a poll right now, so if you have any questions, feel free to input them now. While we're getting some questions, maybe I could start off with one. So thank you, Dr. Sato, for specifically going into the GPCR library, as well as to the TAO optimization. I know on one of your final slides, it was very brief, but you did mention other libraries. So could you just maybe go in a little more detail, what other libraries are accessible now through our collaboration?
Yeah, thanks, Jordan. So we are creating a fully human Fab library as well. We call it the Hyperimmune library. That'll be a library that we can utilize for discovering antibodies to any target, not just GPCRs. And we're also, that library is available. We're also making a whole series of VHH single domain libraries that are based off of llama alpaca diversity. And so, and those are also been validated as well. And so those 4 libraries will now be available through our collaboration with LakPharma. And we plan to create more, as I indicated, throughout the upcoming months to come.
Great. So we have one question in whether Twist library is suitable only for GPCRs or all targets.
Yeah, I think I kind of just answered that through talking about what we're going to do in the future. So this initial library that we created is really just for GPCRs, but we definitely plan to create, and have already done so, a whole family of different libraries that can be used for any target.
All right, thank you. I think that's all the time we have. Thank you again for joining us from across the globe. And if you're interested in learning more, there's further questions and an email that will be sent out shortly. Have a good day.